Sedetevi che cominciamo. Un momento, un attimo. Beatrice, scusa che sorry, do we have to start? Okay, thank you very much. Thank you for coming back. I'm very happy to start this afternoon session with our roundtable. Maybe those of you who were here also last year know that this is the day where we always organize this roundtable. We started with this initiative during the COVID, and then we are continuing, and why to have a roundtable? So the roundtable is a different scheme, with respect to the classes or the lessons that you have experienced so far. So roundtable means that we should have a roundtable discussion. So as I was saying this morning, we are truly looking forward to discuss with you a topic that to me is very appealing. As every year, I discuss with the director, Professor Macchia, that you know here, and we try to pick the most interesting topics. So at the beginning, picking a topic during the COVID era was the COVID, drug discovery for COVID, and so on. And then this year, which is my last year, I have to say, maybe I should introduce myself, sorry, before, because the community, the Italian community knows me, and but I'm the president of the Division of Medicinal Chemistry of the Italian Chemical Society. I'm almost finishing. I will finish at the end of the year, so my term is almost done. And we will have election, so we heard about the election even yesterday, by Isabella, so I remember to vote, to express your vote for the election. But now, this was for the Italians, but now let's switch to the topic of this roundtable, this year roundtable. And so it was my last year, and I was sitting and say, what should I do with this last year topic? And I wanted to look ahead, because you know that I like to stay here, to participate, to everything with you, you know that. And so let's take a look to the future. So that's why the title of this roundtable this year is The Future of Medicinal Chemistry. Okay, we could have talked about the future of medicinal chemistry in a free way, Beatrice is here, but she is representing the European Federation of Medicinal Chemistry. There are so many renowned speakers, and so we could have discussed ourselves among ourselves. But to me, the best way to look at the future, in a bright way, is counting on you. It's counting on you, and to what you can bring to our beloved discipline, okay? What you can bring with your mind, with your passion, with your dedication. And so I decided that to talk about the future of medicinal chemistry, I wanted to hear the voices of young scientists. I mean, not like you, PhD students, but not as old as me, okay? So that's why I selected those I considering the emerging voices. What does this mean, emerging voices? In my opinion, the emerging voices were those scientists who are considered the rising stars. We know that there are not just these sitting at this table all connected abroad, but I wanted to touch specific topics And for touching specific topics, I try to select what to me, and not only to me, because they have been recognized by the community with several awards, I wanted to talk with those that are the emerging voices. So I don't want to spend other times. I wanted to share with you the structure of the round table. So I believe we can start. So the first, at the beginning, to introduce the topics, the future direction of medicinal chemistry, all the speakers of this round table will make a presentation. I'm not going to go into details too much in their career, because they will go to present themselves, and everyone will touch a specific topic, you will see. Then after the presentation, we will start the roundtable discussion. I have received several questions by you. I'm truly glad for that. And so very thankful, very grateful. So we'll start with this question, and then we will open the discussion to everyone. So we can start with Chiara. Please, Chiara, if you want to come. Chiara does not need introduction because you have already met her. I just want to highlight that Chiara has been awarded in 2022 by the L'Oréal UNESCO Award for Women in Science. So let's see what is and the topic that we discussed and we agreed on is allostery, that we have not touched allostery. So let's see if you consider allostery as a promising direction. Thank you very much, Chiara.
Thank you. Just wait 1 second that we are trying to share also with the perfect. Okay. Can I take this one?
No, because it's not
okay.
working.
Yep. Okay, so in the next 15 minutes, I'll try to convince you that allostery is one of the topics of medicinal chemistry in the future. But before starting, we already... How can I go? Yeah. Okay, so before starting, I'd like to briefly mention my career path, even though we already met over the last 3 days, as maybe can be of inspiration of any of you guys. So I studied my undergraduate at the University of Modena, and then I moved for my first period abroad at the Montfort University of Leicester. And then I started my PhD in medicinal chemistry at the University of Modena as part of a European project that was involving scientists from all over the world. And during my PhD, I did 2 visiting periods, one abroad, one at the National Learning Research Foundation in Athens, and one at the University of Albany in the United States. And I also joined the visiting period in the lab of Maria Laura at the University of Bologna. So I'm sure you will hear a lot also from the other speakers. It's really a suggestion from all of you to really go abroad, go in other labs in Italy or abroad, and expand your knowledge. And then I wanted to do academic career, so I moved to the University of Basel for my postdoc, where I stayed for more than 5 years. And then in 2022, I wanted to come back to Italy, and thanks to 3 fellowships, I started my independent career at the University of Milan. Okay. So before talking about allosteric compounds and allostery, I'd like to briefly mention the difference between orthosteric compounds and allosteric drugs. So you might know that orthosteric compounds are molecules that bind to the active site. So the active site is the most obvious pocket within a protein. And then orthosteric drugs bind to the active site, prevent the substrate from binding, and we do have a therapeutic effect. So most of the drugs we have in the market are actually orthosteric inhibitors. But there might be some problems. A problem is that the active site is very similar. So for example, if we think about kinases, and Matthias is going to give you a nice insight on this, but just briefly, there are more than 500 kinases in the human genome, and they all bind to the ATP. So you can think that the ATP binding pocket is really similar. So if you want to bind to the active site, it's very difficult to really achieve selectivity. And this will lead to multi-target compound, multi-kinase inhibitors, and severe side effects. Another possible disadvantage is that orthosteric inhibitors lead to a complete inhibition or activation rather than modulating the protein function. And I will show you later that allosteric compounds lead to a modulation of the function of a protein. Okay, but what are allosteric compounds? So allosteric compounds are molecules that bind to a site, a secondary site, and they transmit the signal to the orthosteric site and lead to inhibition or activation of the protein. So we can have allosteric inhibitors or we can have allosteric activators. What I'd like to remind you that is not enough to bind to a secondary site in order to say I have allosteric modulation. You have really to transmit the signal to the active site and modulate protein function. If you are only binding to a secondary site, you might have just a ligand. And allosterism was defined more than 50 years ago by Monod and Jacob as the second secret of life. And even though we are studying on this, and researchers are studying on this, it is still one of the greatest unsolved mysteries of protein function. It regulates all protein, and this is really an intrinsic property of all protein, and regulates all biological processes. It can be used to control enzyme, G-protein coupled receptor, and ligand gate ion channel. And I'm going to give you a few examples today about enzyme and G-protein coupled receptor. So you might remember from my talk that I'm interested in PI3K alpha, and I'll show you covalent inhibitors of PI3K alpha. And I want to share with you this paper that was published last year on allosteric inhibitor of mutant PI3K alpha. There was actually a nice question from some of you guys asking how we can selectively targeting only the mutant isoform. And this was a nice paper where they developed an allosteric inhibitor targeting a previously unexplored pocket. And they showed that the compound worked in animal models, so the tumor volume versus the day of treatment, you can really see that this compound in red reduced the tumor volume. And the inhibition of mutant PI3K alpha really allows to limit the metabolic side effect that I mentioned to you the first days, targeting PI3Ks. If you have a pan-PI3K inhibitor, you have hyperglycemia and hyperinsulinemia. So here they develop compound targeting only the mutant PI3K alpha and they minimize the side effect. And now this compound is in phase 2 clinical trial for the treatment of solid tumor. Okay, so most of the time we want to inhibit kinases, for example, but we can also activate them. And then here is a nice paper published in Nature last year. So this is also to give you an idea of how important is becoming allostery. All the papers that now are publishing allosteric compound are really published in Impact Factor Journal. And this is a paper that have been published in Nature last year where they developed allosteric activators of PI3K alpha. And they found a pocket that is only present in PI3K alpha. You can see here that you have a steric clash in beta and delta. And the author showed that this compound lead to cardioprotection and could be exploited in patients with stroke or myocardial infarction. And it also stimulates nerve regeneration, and for this reason, activation of PI3K alpha opens novel opportunity in drug discovery, for example, for cardioprotection, neuroprotection, and neuroregeneration. They have also proved that actually a short and transient activation of PI3K alpha is not leading to tumor, because of course I've shown you that we want to inhibit PI3K alpha in order to have an anti-cancer effect. So, but they show that short time and transient activation can be beneficial. And we discussed about chemical probes. Of course, an activator, a selective activator, can also be used in chemical genetics in order to really explain the role of this protein in organism or disease model. So if we think about allostery, another example that we should mention is KRAS. So KRAS for many years, it has been considered undruggable. And this because KRAS is a GTPase, so it's different from, for example, kinases that they bind ATP. ATP can be displaced with orthosteric inhibitors. On the contrary, GTP has a picomolar affinity for GTPase has picomolar affinity for GTP, so GTP can't be displaced. And for this reason, for many, many years, KRAS has been considered undruggable. But it has been discovered a pocket that can be exploited for covalent allosteric inhibitors. And they develop a compound that you can see here in the figure that is binding to KRAS G12C. So you can see here the cysteine, the compound is covalently binding inside this allosteric pocket. And this result has been published. KRAS has been really drugged by the group of Kevin Chokat, and it has been published again in Nature more than 10 years ago. And now, only after 10 years, actually, this compound that you remember, we discussed this at the workshop, is a covalent allosteric inhibitor of KRAS, has been granted approval for the treatment of non-small cell lung cancer. So it really went to the clinic. So from being undruggable to getting a compound into the clinic in almost 10 years. So this is really an excellent achievement. Then if we think about allostery, we also have to mention the G-protein kappa receptor. So the G-protein kappa receptor is really a huge family and is one of the primary targets for marketed drugs. If you think more than 30% of the drugs currently marketed in the United States are binding to GPCR. And there are many allosteric pockets that can be exploited, and there are many diseases that can be treated with allosteric modulators of GPCR, from cancer to neuropathic pain to viral infection. And as I mentioned before, allosteric modulators can really be positive allosteric modulators where they increase the signaling, negative allosteric modulators where they decrease the signaling, but we can also have neutral allosteric ligand where the signaling is unchanged. So as I mentioned before, it's not enough to just bind there in order to have a modulation. Actually, for GPCR, it has been pioneered the concept of bitopic compound. So bitopic compounds are molecules that present in their scaffold two pharmacophores. So one part binding to the orthosteric site and one other binding to the allosteric pocket, and they are connected through a linker. Here you can see some example of bitopic compound of the muscarine acetylcholine receptor M1. You can see in green the allosteric pharmacophore, in blue the orthosteric pharmacophore, and in the middle there is a very short linker. This is because these two sites are very close by in GPCR. So once the concept of bitopic, bivalent has been pioneered with GPCR, it has been used also for other protein and enzymes. And this is an example of a bivalent molecule that has been developed on mTOR, that is a kinase. And you can see here in red is the ATP site. And in blue, in cyan, is the allosteric pocket. And you can see here that they are very far away. But the group of Kevin Chokat, and this also was published in Nature, published this compound where they were connecting the active site inhibitor with the allosteric site inhibitor with this very short, very long linker. So if you look at this molecule, you will immediately recognize the huge molecule, and you will think that it's not cell permeable. And actually, this is really cell permeable. It really works in cell. It works also in xenograft models, so it was really a great achievement. And the bivalent compound actually allows to inhibit also the resistant mutants. So this compound was active in wild-type cell, but also in cells that acquire resistance to both orthosteric and allosteric inhibitors. So really, the bivalent compound can be used to counteract resistance. Okay, then before concluding, I'd like to go with you through the potential advantages, but also the challenges of allosteric modulators. So first of all, we said that the allosteric pockets are less evolutionary conserved. So these usually are present only in one protein within a family, and these really allow to develop compound with excellent selectivity, and so to limit the side effects. in addition, we can selectively target mutant protein. And this, for example, the example I've showed to you before, where we can selectively target mutant PI3K alpha or KRAS. And then we can exploit all targets. So there are some targets that have been exploited for many, many years, binding only to the orthosteric site. We can really exploit all targets, but with new molecule binding to novel pockets. We can combine, we can have combination therapy with orthosteric drugs and allosteric compound. We can give the two drugs at the same time to the patient, but we can also develop, as I mentioned before, bitopic or bivalent molecules. And then we can target proteins that have been for a long time considered undruggable, as KRAS. And then, so here is a nice representation that explains the difference between orthosteric compound and allosteric molecules. You can see that the orthosteric compound are on-off switches, while allosteric compound are dimmer switches, so they allow to fine-tune protein functional activity rather than causing a complete inhibition or activation. And this, of course, limits the side effects. Okay, but there are also some challenges. So first of all, the allosteric sites usually are smaller than orthosteric pockets, and then it's difficult to find inhibitors, find compounds that binds there. And most of the allosteric compounds that we now have, either in the clinic or in drug development, they have been discovered by serendipity, or they have been discovered by time and resource-consuming screening campaign. So it's very difficult to rationally develop allosteric compounds, and the success rate of allosteric drug discovery is very low. So it is estimated to be 1 over 10,000. And then minor differences in allosteric modulators can lead to opposite effects. So this you have really to pay attention, because it could be that only changing one atom within a compound then leads to an inhibitor to be an activator. And then allosteric sites are often unknown and difficult to predict. So I'd like to leave you with a question, actually, if we will be able in the future to rationally and systematically design allosteric modulators. So thank you for your attention, and I'm then happy to discuss with all of my friends and colleagues.
Thank you very much, Chiara. Now we will move to the second speaker of our roundtable, which is Matthias Geringer. Matthias is in Tübingen. Are you in Tübingen? He works in Tübingen. I'm not sure, Matthias, whether you are in Tübingen at the moment.
No, at the moment I'm on a
research
retreat at a monastery somewhere in the countryside, but it seems the internet connection is good.
Okay, so Matthias has a long track record of achievements, but I'm biased. I have to confess that I'm biased, but I want, you are laughing, Matthias, already, you know what I want to say, maybe. But I want to highlight that this year he has been awarded with a Phil Portuguese lectureship by the Journal of Medicinal Chemistry, ACS Medicinal Chemistry Letters. I'm biased because I was trained by Phil Portuguese, and so to me it is a particular honor to have Matthias here today. And because we had already a session dedicated to covalent inhibitors, we want to hear from Matthias with the future direction regarding covalent inhibition. Thank you, Matthias.
Yeah, thank you, Maria Laura. So I'm trying to share my screen. So 1 second. Now you should be able to see my slides and hopefully my pointer
very well.
as well.
Yes.
The pointer as well?
Yes.
Okay, perfect. Then let's start. So yeah, welcome to my short talk on what's coming next in the field of kinases and covalent inhibition. So I also want to give a very brief introduction of myself. Actually, I am a chemist by training. I was studying chemistry at the universities of Heidelberg and Karlsruhe, and also for some time at the École Nationale Supérieure de Chimie in Montpellier in southern France. And after that, I moved to Tübingen, where I did my PhD thesis in the group of Stefan Laufer. And actually, after that, I moved to Switzerland, to the ETH in Zurich. And there I did a postdoc with Karl-Heinz Altmann in natural product total synthesis before coming back to Tübingen, where I was first a junior group leader, then promoted to an assistant professor. And very recently, I was recruited to the Tübingen University Hospital, which is also part of the University of Tübingen, as a full professor in medicinal chemistry. So I guess you all know about protein kinases. Protein kinases are enzymes that transfer a phosphate group from ATP onto a hydroxy group on proteins, either on a serine or threonine residue or a tyrosine residue. And actually, this phosphorylation is a post-translational modification that is reversible, that can be reversed by protein phosphatases. And together, protein phosphatases and kinases act as a kind of on and off switch for protein function. Therefore, protein kinases have very important roles. They are essentially the backbone of intracellular signal transduction, and their dysregulation also drives a lot of diseases, especially cancers, but also other diseases. The human kinome is relatively complex. It was first assembled in such a phylogenetic tree by Gerard Manning, and it contains over 500 protein kinases. And amongst those are a lot of kinases that have already been drugged. I will show that on the next slide. But there's an even much larger fraction where we have no selective inhibitors. For some kinases, we don't even know what they do. This is the so-called dark kinome. And researchers agree on that. There are still a lot of interesting targets about to be discovered in this dark fraction of the human kinome. So if we talk about covalent kinase inhibitors, what we are essentially talking about is what we now call targeted covalent inhibitors. And these are compounds that consists of a reversible recognition element that must bind the target of interest with a certain affinity and also a certain selectivity, and thereby put a reactive group, typically a low reactivity electrophile, which is the covalent warhead, in close proximity to a nucleophilic target amino acid. And only once this warhead is brought in close proximity here, it should trap the target amino acid, usually in an irreversible manner, but this can also be reversible. So if we look at the current landscape of approved kinase inhibitors, we see there's a lot going on, and most of these compounds are approved for cancer indications, which is highlighted in green on this slide. But as we can see here, in the last years, other indications are also emerging, and there are also nowadays already a couple of covalent protein kinase inhibitors approved. It's 10 at the moment, and I'm sure more will come. So far, covalent kinase inhibitors are mostly used for cancer, but I think this is one of the things that is about to change. So in the past, people were quite afraid of covalent targeting approaches because they thought this would cause severe side effects and idiosyncratic toxicities. But it turned out that it is quite controllable. And for now, in the last years, there was kind of dogma that people said, okay, we can do covalent approaches, but only for really severe diseases like cancer, which are life-threatening. And I see some change, at least that more and more covalent approaches are also being employed to indications beyond cancer. Just to give you two examples, for example, there's a compound called ritlecitinib. This is the first covalent kinase inhibitor approved for non-cancer indication, and that inhibitor targets a protein kinase called JAK3. JAK3 is a kinase that controls immune response in a relatively selective manner, while the function of the other Janus kinases is much broader. And there's strong genetic evidence that selective suppression of JAK3 function, leads to immunosuppression at limited side effects. So people try to make selective inhibitors, but this is not trivial because the Janus kinases are very similar, but they have a tiny little difference in the hinge region, which is in the front pocket, which is a cysteine in JAK3, which is a serine, a serine in the other JAK kinases. And so researchers from Pfizer who developed this compound, a pan-JAK inhibitor called tofacitinib, used this compound as a starting point actually to address the cysteine 909 in a quite interesting design approach where they removed this methyl group here, which shifts the equilibrium from a syn-like to a more anti-like conformation, which is then more preferred than in that compound, and thereby orients that residue to the other side here. And with a little bit of optimization and fine-tuning, they finally came up with that compound, which you can realize is relatively similar to that one, which is an approved covalent inhibitor. You can see the covalently bound form there. It's ritlecitinib. It has very efficient inactivation kinetics in the range of 10 to the power of 5 per molar per second. Potent IC50 and especially important, very selective against the other JAK isoforms. It's inhibiting some other kinases which have a similar cysteine, I have to say, but it's highly selective here within the family. And that compound was approved in 23 for alopecia areata and is currently under clinical trial for other autoimmune indications. Another example is remibrutinib, a compound being developed by Novartis, which is currently also in clinical trials. And that compound targets a kinase called Bruton's tyrosine kinase. And that kinase is known as a target for covalent kinase inhibitors since long because it's a target for blood cancer as well. But you can also address this kinase for targeting autoimmune diseases. And this is what these people around Daniela Angst wanted to do. And actually, they developed that compound with a relatively unreactive warhead and this moiety binding a special pocket in BTK, which promotes selectivity. And by that means, they were able to generate a compound that is highly selective in the family, way more selective than the covalent BTK inhibitor ibrutinib, and at the same time this compound achieves what we want to have usually for covalent compounds, which is a disconnection of pharmacodynamics and pharmacokinetics. So you can dose the compound, it hits the target, gets metabolized rapidly, but the effect, as you can see on the right, stays because the target, once covalently inhibited, stays inhibited and has a relatively low turnover, so you have a prolonged effect. And this is actually one of the things that we in many cases like to see for covalent inhibitors. And that compound, very potent, very selective, suitable for oral application and currently in clinical trials for autoimmune diseases. So another thing that I think is coming up more and more is the use of non-standard electrophiles for covalent targeting. And this is something my personal research is also pretty much dedicated to. I've also recently published a perspective article, quite comprehensive one, together with my group in the Journal of Medicinal Chemistry, where we summarize all the developments in targeting cysteine, but also other amino acids with non-canonical warheads. So if you're interested in this topic, you can check out this article and its previous version from 2019, and you will have a quite comprehensive overview. So if we look at the currently approved covalent protein kinase inhibitors, we can see that all of these compounds have alpha-beta unsaturated amides as the covalent warheads. These alpha-beta unsaturated amides, they are attacked by the cysteine in a so-called sulfur or thiamycyl addition to form such a relatively stable beta-thioether adduct. But of course, there's much more chemistry that we can use in that context. For example, carbonyls with alpha-leaving groups, strained heterocycles or even carbocycles, electron-deficient aromatic systems with a double or triple bond attached that can act in a Michael-type fashion, or what is coming up more and more are covalent compounds or warheads acting via nucleophilic aromatic substitution chemistry. These are electron-deficient arenes or heteroarenes with a leaving group where the cysteine or possibly also another amino acid that is nucleophilic can then attack, kick out the leaving group and form a covalent bond. And that has been done, for example, in this compound here, gamzemetinib, which was developed by BMS, and that compound actually targets the MAP kinase activated protein kinase 2, and that compound went to phase 1 and 2 clinical trials for the treatment of rheumatoid arthritis. And that compound, it's not super potent, but it's already available, and it shows that this is a feasible approach when you aim for a clinical translation. We have also worked in that field. For example, here, this is a compound recently published by my lab, an isoform selective inhibitor of the kinase FGFR4, very potent, highly efficient in activation, rate constant in the same range as many approved covalent kinase inhibitors. If you're interested in that, it's recently published in JMed Chem. So, and I think this is a very viable approach to use such alternative warheads, especially if you aim for targeting buried amino acids, which are a bit deeper inside the pocket. Beyond that, I think reversible covalency is also on the rise in the kinase context. And actually, what many people do if they want to address cysteine in a reversible covalent manner is they use such dually activated Michael acceptors. And these dually activated Michael acceptors, they are more reactive than normal Michael acceptors, but at the end, they are also reversible covalent warheads. We have used that, for example, in that compound developed by my colleague Michael Foster, which is a selective JAK3 inhibitor and a chemical probe. Researchers from Principia Biopharma have developed that compound, which is now being further developed by Sanofi, rilzabrutinib, which is in phase 3 clinical trials. And the interesting thing is, like bound on target, this compound behaves almost like an irreversible kinase inhibitor, but once the target is degraded, it immediately diffuses off. So we can combine like the best of both worlds, non-covalent targeting and irreversible inhibition into one compound if you do it properly. How does that work? Actually, as in the previous case, the cysteine can undergo a nucleophilic attack here to form this beta-thioether adduct. But in that case, due to the 2 electron withdrawing groups, the alpha proton is significantly more acidic, gets easily abstracted to form the anion that can then kick out the thiolate again to regenerate the compound with a warhead. So this promotes the high reversibility in that case. But also there's a lot of other chemistry that you can do. For example, you can use activated nitriles or cyanamides as being done in this JAK3 inhibitor developed by Pfizer. Or you can also use aldehydes, for example. Researchers from Novartis have done that with this FGFR4 inhibitor called oblitinib, which is also a reversible covalent compound that went up to phase 2 clinical trials. Can you also use like such fluorochloroacetamides that give a hydrolyzable adduct that's strictly not reversible since the product is a different one than what you put in, but also by that means you can generate something that is cleaved off from the protein once it is degraded, which can limit side effects. So I think these are just some examples of what's going to happen in the future, but I think there are a lot of other avenues to go. I think it's very important that we go more and more for understanding what's going on in the dark fraction of the kinome, and a significant part of my personal research, but also of other researchers out there, is devoted to that. Then I think we will be aiming towards targeting amino acids beyond cysteine. That's not a trivial task, I have to say, but that opens up a lot of opportunities if you can target tyrosine or lysine or histidine, etc. Then allostery, Chiara has already talked about that, is a hot topic, of course, and that can also be combined with covalency in a kinase context. In general, I think combination therapies in kinase inhibitor research are very important to prevent or to break resistance, which is always a topic, especially if you go for cancer indications. Then kinases or kinase-like proteins that do not have kinase function can also be addressed by compounds that do not inhibit catalytic function but may interrupt other functions of this protein. And there are also researchers which look into disrupting specific downstream signaling pathway of individual kinases. No need to say that degraders are a hot topic, and after some negative experiences in the past, research on targeting protein phosphatases, which counteract kinase action by dephosphorylating proteins, this research is also coming up again. So these are my perspectives on what's coming next in the kinase field. So I want to thank all of you, my funders, and also my group, and of course you for your kind attention. And I'm looking forward to the roundtable discussion. Thanks.
Thank you very much, Matthias. We have maybe already heard this concept in the first day, but as we know, repetita iuvant, we say Latin, so I'm sure that this very nice talk has allowed you to fix some concepts and have a further food for thoughts. And then I would like to move to Luca Larraia. As far as I have understood from the CV, Luca was born in Rome, but then he grew up in Austria. Is it correct, Luca?
Yes, that's correct. I'll say a little bit about that in a second as well, but go ahead.
Yeah, but Luca now is in Copenhagen, and then he will talk about the next generation strategy for the discovery of functional natural product spark compounds. We know very well that natural products have been a rich source of drugs, and they have still a lot of potential. So I'm very looking forward to what is your idea on the topic, Luca. So thanks.
Great. Thank you very much. Can you see me in the correct mode, or am I in presentation mode?
No, it's not in the presentation mode.
So it's, you can see my like
your PowerPoint. I see your PowerPoint.
Yeah, just the slides, nothing else.
Just the slides.
Okay, perfect. Thanks a lot. Good. All right. So thanks a lot, Mariana, for the very kind introduction, and I hope you can all hear me okay. Yes. So I'm gonna, before I dive into sort of what my group and others are doing in the sort of space of natural product inspired and derived compounds, like everyone else, I'll say a little bit about my background. So Mariana already said something about this. I was in fact born in Rome, but my parents moved to Vienna when I was 5. So I did I basically grew up there. But I moved away for an education, university education, first to London, where I did my undergraduate and master's, and then to the University of Cambridge, where I did a PhD in chemical biology. Following that, I moved to the Max Planck Institute.
Sorry, Luca, if I interrupt you, we are always on the first slide.
Oh, okay. That's not great. Just a second.
And if you can even talk closer to the mic, because it's...
Yes. Let me just...
not very clear, your voice. Thank you. Oh.
Thanks for that. Just a second. I will then...
Yeah.
Yeah, look, I think the presentation mode and what you should see on your screen is inverted. So
yes.
Not sure what's going on.
Is that better?
That looks good.
Yeah, that's great. That's what I was referring to earlier, but sometimes it's hard to tell. Okay. So can you hear me a little bit better now?
Yeah, it's fine now.
All right. So I'll jump through this. Yes. So after I moved to Dortmund as a postdoc, and then after that moved to just north of Copenhagen, where I started my own lab at the Technical University of Denmark in 2017. So basically, my group is really interested in natural product inspired and derived compound libraries and sort of their application in medicinal chemistry. So there's a lot of ways that one can sort of think about sort of using natural products. And of course, one way to do this is to take natural products themselves but this can have some issues because of the sort of accessibility of these compounds in a sort of enough scale, that in a large enough scale that you can actually work with these. For those that are accessible, you can either use them directly in screenings, or you can sort of derivatize them and in different ways. In this approach, for example, known as complexity to diversity, where you modify the core scaffold, access a sort of a diverse set of natural product derived compounds. But to take a more sort of synthetic chemistry approach to this, sort of diversity-oriented synthesis was introduced some 25 years ago, where basically in a fully synthetic approach, you use the sort of build, couple, pair strategy, where you synthesize building blocks that can be coupled and paired to access basically compounds that have features of natural products, but don't necessarily sort of directly resemble or have the natural product scaffold. So I think where people have come, you know, in the last 5 to 10 years, is to sort of merge these strategies a little bit, where you take natural products as the sort of guiding principles of, you know, towards the library synthesis, and in one approach, you sort of simplify these to their constituent scaffolds, and then which you can then access synthetically, and then access essentially sort of natural product analogs by synthesis in this biology-oriented synthesis approach. The limitation here is that you always focus on the scaffold that was originally found in the natural product. So in a more recent approach, pioneered in the Waldman lab when I was there, we basically sort of fragment natural products into their constituent fragments, which can then be recombined chemically to access sort of what we call pseudo-natural products. And these are sort of natural product fragments recombination that we don't sort of see in nature itself. So we can sort of access a sort of a larger part of chemical space and new chemical structures. So where we come into this, and when we go about embarking on a project like this, you know, it really depends on what the biological rationale is behind behind your approach, essentially, or which one you want to choose. So to give you an example of, for example, what my lab is doing, we are really interested from a biological point of view in a family of proteins known as cholesterol transporters. And I'm not going to go into detail about these because it's beyond the scope of this talk, but it's enough to say that there are three families, the ORPs, the STARs, and the ASTERs. And these are different in a little bit in their structure, but more importantly in their function. So they can basically transport cholesterol between different membrane-containing organelles in the cell. And so each member of this family has a specific role, and in sort of transporting cholesterol between specific membranes in a cell. And studying these and targeting these proteins from a medicinal chemistry point of view, it comes with a lot of challenges, which also one of the things that makes it interesting for us. So, for example, from a biological point of view, there's actually quite a lot of functional redundancy within this class of proteins. So, for example, when you knock one of them out, another one can kind of sort of step in and take over its function. So it can actually be very difficult to understand the specific function of these proteins just using genetic methods. From a small molecule point of view, there's actually, at least when we started this approach, there were very few known inhibitors, and those that are available are either highly complex natural products, such as reterazine B, which can be made by total synthesis, but it's 50 steps, and you get sort of half a mg out at the end. Or you have very low selectivity compounds, such as U1866A, which is an extremely non-selective sterol transport inhibitor. And so the challenges there in terms of selectivity come from the structural similarity of these proteins. They all bind cholesterol. They all share some similarities in their sterol binding domain. So our approach to tackling this is to take basically a combined approach of these various compounds library synthesis strategies, which sort of looks like this, where we can take a sterol-derived primary fragments from different types of sterols with different A and B rings. And then this should provide a sort of called affinity handle that sort of should be more easily recognized by the proteins and by the targets. But then we can fuse them synthetically to diverse secondary fragments, which should provide the selectivity for a given sterol transporter over another. And we have some criteria when we make these compounds. We try to keep the step count down, try to access scaffold fusions that have not previously been sort of observed. We also have seen in our work that 3 to 5 analogs per combination is both necessary and sufficient to actually get biologically active compounds. And we make the compounds in a racemic fashion, which I'll come back to a little bit later on. So just to give you a quick example of the kind of chemistry that we do, so we use kind of ketones as these as useful functional groups here to access a range of different scaffold fusions. On this slide, it's either sort of very classical heterocyclic chemistry to make indoles, quinolines, aza-indoles. We can also focus on some spirocycles and some ring expanded products over here. So ketones are really good building blocks for this kind of chemistry where we can access different sort of scaffold fusions. But of course, from ketones, you can also make some derivatives, such as these hydroxymethylines, which can then be used to access different isomers of pyrazoles and pyrazolopyrimidines. And while from a synthetic point of view, it might not be so desirable to make, to get two different isomers in each chemical reaction, from a library point of view, this actually becomes in quite handy as long as they're separable. I'm going to show you some examples of why this is also useful biologically speaking. So, in addition to these kind of what so-called edge-fused kind of scaffolds, where we have bolted on a sort of secondary fragment onto a first, we can also target some different sort of library synthesis approaches. And the first idea here is that we have now moved from a transdecadent system found in in certain steroids to a cisdecadent system, for example, found in cholic acid. And we can make the same edge-fused analogs on this sort of top row in red and blue. But we can now also apply a sort of a complexity to diversity ring distortion strategy to access additional scaffolds that we haven't really looked at before and introduce a second sort of level of diversity in the compound libraries that we make. So what does this look like in practice? So if we start with this kind of indole-fused cisdecadent system, you can actually use a range of different oxidative conditions to access 5 new different scaffolds. So we can use, for example, an oxidative ring expansion here to go to the keto lactam, which can then be recondensed to give a quinolone. We can also use sort of oxone instead of sodium periodate here to access this hydroxy indole. And then we can also, by varying the amounts of oxone and the solvent, we can actually go all the way to this sort of spiroxindole type product. And finally, we can access this very interesting looking scaffold here, which I'll show you the mechanism for in just a second, which is sort of a more unexpected result in this work. And just to point out that we have really made these compounds, we do have crystal structures of all of these, just to highlight that sort of spatially and structurally, these are really quite different looking compounds at the end of the day. So that sort of fulfills sort of chemical and structural diversity criteria that we like to see. So this, just to show you the mechanism of this kind of unexpected reaction, we actually see initially a bromination of the indole, followed by attack of water here. And this undergoes a semi-pinnacle rearrangement to access the spiroxindole shown here on the right. And this is actually the compound that we sort of initially targeted, but actually this reaction has acid present. So basically, what happens is you protonate this lactam. And if you look at this compound in 3D, you can have a sort of intramolecular condensation reaction, which allows you to lose water and give you this oxypino-indole type structure here. And we'll come back to why this is interesting in just a minute. So we've made a lot of compounds. We made actually over 700 compounds of this sort of sterile-inspired library. And of course, that in itself is not interesting unless you kind of test them. And here we come back to kind of the assays that we use for all of our sterile transport proteins. So we've recombinantly expressed and purified 10 different transporters. And for all of these, we have both thermal shift assays, where you can see basically, you can measure sort of a melting curve for a protein, and this is typically stabilized by a ligand. But we can also use fluorescently tagged cholesterol derivatives to look at it as fluorescence polarization-based displacement assay with an inhibitor here, where you can see a change in the retention of plain polarized light in the presence or absence of an inhibitor. So what we did is we initially screened the first proof of concept library, which at the time was only 65 compounds, in a thermal shift assay. And we identified both inhibitors, or rather ligands that stabilize aster-C, so in this top row over here. And we call these astersin-1. And we also identified some stabilizers of aster-A. So these are 2 different proteins of the aster family of sterile transporters. And immediately you can see that they both contain the same A and B ring scaffold, but they have differences in the secondary sort of fragment that is fused to the steroidal fragment, and that sort of provided us an early hint that the strategy seemed to work. As I mentioned earlier, we make all the compounds as racemic mixtures, and this was the selectivity of astracin 1. So it was moderately potent and reasonably selective compared to aster B and C. But actually, when we synthesized both enantiomers, we saw that that unexpectedly the enantiomer with the unnatural stereochemistry, so the one that doesn't match cholesterol, was actually both more potent but also more selective compared to the natural one. So if we had made everything as an anti-selective synthesis with the natural stereochemistry, we wouldn't have found basically this interesting result. I also mentioned that we had this very interesting sort of ring contracted and cyclized compound over here, and this actually turned this series of compounds actually turned up as hits in our aster screen, and very surprisingly, in fact, this compound, which we called asteroxin 1, was the most potent aster A inhibitor reported to date. And we also have some SAR for this series. We've made a lot more compounds for the scaffold since then, but in general, they all share the same feature, that they are fairly potent against aster A, but essentially have next to no activity against the most sort of homologous proteins, aster B and C, and also very weak inhibition at 10 micromolar here of other sterol transporters that we also tested. So these compounds look, they look pretty potent, but most importantly, they look really, really selective for aster A, which we kind of wanted to get to the bottom of. I mean, these compounds don't really, they don't look steroidal anymore. So how do they really bind to our target? So we don't have a crystal structure, unfortunately, yet, but we have docked these compounds into aster A, and we see quite interesting results here because actually we can see that the aster A binding site, even though it's designed to bind cholesterol, which is actually a fairly flat and rigid molecule, seems to have a little bit of three-dimensionality to accommodate this quite sort of large sort of ring system at the base of the pocket here. So we think that the selectivity comes from the fact that actually the binding site is a little bit larger and can accommodate this, whereas other sterile binding sites may not be the same. And we also are predicted to pick up a key hydrogen bond here towards the exit of the binding site. And apart from that, we have quite a lot of sort of hydrophobic interactions going on. So actually, we had this model which predicted that the minus enantiomer would be the most active, and then we actually synthesized both that and the other enantiomer, and it turns out that, as predicted, it was in fact the compound with the unnatural stereochemistry that turned out to be the most active. Even though arguably this compound does not resemble a sterol anymore, so the concept of natural and unnatural stereochemistry is maybe less relevant here, but it again supports the fact that making these libraries as racemic mixtures initially, we may uncover some unexpected bioactivity based on these compounds. So with this, I will close and thank my group for all their hard work. And I wanted to just finish up by apologizing that I couldn't be here in person. So we have an honorary group member over here, which is my daughter. And there's another daughter hopefully coming very soon, which is why I couldn't be with you in person today. But I hope to meet you all at some other point in time. So thank you.
Thank you, Luca. So we are waiting for you in Urbino with all the family next time in person.
Hopefully.
Okay. I also forgot to mention that Luca was the recipient of the RSC Emerging Investigators in Medicinal Chemistry. So anyway, thank you for showing the beauty of your chemistry of these and the beauty of these molecules. And now we will move to another truly emerging topic, which is PROTACs, and we have the pleasure of having here Andrea Testa. Andrea Testa was started to work in PROTACs with Alessio Ciulli, and now he's working in Anfista in Oxford, in this company dedicated to PROTACs, and he will talk a bit about his career, but I want to mention that he received in 2022 the Talented Young Italian in the UK's award. So we are very proud of you.
Young is the most important word.
Young, yes. But we are very proud of you as Italian. I'm very proud of you. Thank you.
Thank you, Maria Laura, for the invitation. So yeah, it's a pleasure to be here. With this talk, I just want to give you a bit of a perspective and maybe an introduction, although I saw that quite a lot of people already worked in PROTACs from the posters that I had the pleasure to look. So a little introduction on targeting 3 ligases for inducing target protein degradation, and PROTACs, glues, and probably where the future is going to be, which is something in between there. How do I change it? Oh, yeah. This, okay, that's fine. Yeah, so as everyone else has done, just a little bit of my path to degradation, so that how I end up doing what I do. So I'm originally from Bergamo, and I studied chemistry at the University of Milan. Basically training as a synthetic chemist in organic chemistry. And then I moved for my PhD to the University of Aberdeen, and the very top of Scotland, where I did a PhD in medical imaging, which was basically radiochemistry and design and synthesis of PET tracers. So very different from the traditional chemistry that I used to do in Milan. And after that experience, I decided the time to stay in Scotland. I really liked there. And I found a postdoc opportunity in the group of Alessio Ciulli before he was famous, I shall say. I joined the group in 2015. At the time, the group was really starting working on protein degradation, and it was really, really exciting. So I think it was on the situation which was the right place at the right time. During my postdoc there, I had the opportunity to, together with Alessio and another postdoc, to spin out a company, which is Amphisa Therapeutics. So we moved out from the university with the company. At first, it was only 3 of us, and now it's about 50. And so we moved from Dundee to Glasgow, because that's where we found our first space for the lab. And after 3 years in Glasgow, we moved to Cambridge, where it's the place to be if you work in biotech. So that's just a brief history. I think I don't know if the slides has been presented before this meeting, maybe not. So excellent, I have the chance to go with that. So this is a bit of the enzymatic pathways that lead to target protein degradation. So with PROTACs or degraders in general, we hijack the last step of the ubiquitin signaling. And that means that we can make compounds that bind to an E3 ligase, and they function like adapter, basically creating a new interaction between the E3 ligase and the target protein that we want to degrade, so that this complex is formed, and the E3 ligase starts to place ubiquitin on the target protein. And this is really at the end of a multi-step process. And we just, by acting at the very last step, we can then very selectively degrade proteins. The ubiquitinated protein is then recognized by the proteasome and is then degraded. So with small molecules, we can hijack this. And all of this has been possible really because of the advancements in medicinal chemistry, especially on targeting E3 ligases. So most of the compounds now in clinical trial, actually all compounds now in clinical trials, relies on 2 specific E3 ligases, VHL and cerebron. So the ligands for these E3s have been discovered actually in a very different ways. For VHL was very much a fragment-based and structure-based design of a peptidomimetics that mimics the natural hydroxyproline interactions with VHL. So we have compounds like this that have been really the basis for the development of PROTACs in academic labs, in industry, but now really in the clinic. And for cerebron, instead, the discovery was much more serendipitous. So a lovely small molecule with a terrible history, but thalidomide and lenalidomide derivatives and POM as well. So they bind to cerebron, and they can modulate the surface of cerebron, so then other protein can interact with cerebron and then get degraded, or they can function as a handle for us to attach linkers and conjugate conjugate protein binders for the proteins we want to degrade. So all of this was really possible. The clinical development of these compounds was possible because of the development of high-quality small molecule ligands. So we're talking about protein degradation, and why is this really revolutionary? Well, that's because inhibition and degradation are two very different processes. So when we inhibit a protein, as Chiara really explained very well before, we need to go into an active site or an allosteric site, but we need to really saturate this binding site, and the compound must elicit a function on the function of the protein. So it must inhibit or cause a rearrangement of the protein structure so that the protein then becomes inactive. It is an enzyme, or we need to block protein-protein interaction at specific sites. So it's really important to develop compounds that have a function. They do not only bind to the proteins. And of course, we need to saturate this, and the saturation must persist if we want to achieve the effect. If the protein is also as a scaffold function, yes, we can maybe inhibit protein-protein interactions, but the protein is still there. So even if inhibited, they can still have some effects. In protein degradation, which is now an event-driven pharmacology, we need to engage the protein that we want to degrade with the A3 ligase, for example, protein B here. And then once it is engaged, we form a ternary complex, ubiquitination happens, so the process then is catalytic because the compound remains inside the cells. The compound doesn't really need to bind into an active site, can be an allosteric site, can be a ligand for any site of the protein, as long as you can form this complex and recruit the protein to the A3 ligase. And of course, then the protein is removed from the cells, so we can have a very different pharmacology compared to inhibition. Proteins and glues are really the kind of modalities that have been used so far for protein degradation, and I'm sure there is a lot of debates on when a protein is a glue and when a glue is a protein, or what is in the middle. But for me, I like a simple definition, which is that a protein really binds to independent and specific binding sites of the A3 ligase, like in this case, we see VHL, and the target protein. So two well-defined binding sites. There is measurable binding affinity for both sides, so we can measure binding affinity at the binary level for the target and binding affinity at the binary level for the A3s. These compounds can be then quite rationally designed once we have the two binders, and we just need to connect them with the right linker. Of course, because they start from two independent moieties, each shown with a good binding affinity, they have a certain complexity. So the molecular weight and size of these compounds tend to be quite larger than the usual drug space. And of course, we cannot degrade, or it's very difficult to degrade, proteins that are unligandable. We need the binding site to on both proteins. So if we do not have a binding site on the protein that we want to degrade, we'll probably never manage. Molecular glues, on the other side, sometimes are considered monovalent, just because they don't look like bivalents, basically, and they bind to a binding site that is formed in between the protein of interest and the A3 ligase. Normally, they have measurable binding affinity only for one site, and normally, if we think about cerebron glues, they have all binding affinity to cerebron, but they do not have a measurable binding affinity at the binary level with the target protein. And because the compounds bind in the pocket between the two proteins, we don't really need a high binding affinity to the target protein. Actually, it doesn't exist very often, and the target protein doesn't need to have a druggable binding site, because what we are going to do, we are really drugging the pocket that is built at the interface. So for this, the rational design is really challenging, and we really need to have a lot of screening capabilities to identify compounds like this, which normally are a bit easier to develop than proteins because they have a smaller size. What is really interesting to me, and I think will be something that in the next few years we'll see more and more, is the compounds that behave with properties that are in between PROTAC and molecular glues. And here I have an example that has been published by the Chuli group in Nature just a few months ago. And basically what was discovered here is a bifunctional compound that binds two domains of the same protein. So here we have BRD4. BRD4 has two domains, two bromodomains, which are highly druggable. And the group has discovered a series of compounds that can bind the two bromodomain sites with one site with high binding affinity, the other one with less, or depending on which compound, even with high binding affinity and high binding affinity. And by binding the two domains of the protein, a structural rearrangement is achieved. And at this point, the surface of the protein, the surface of BRD4 changes because the domains get close together, but also because of the molecule that now sits here. And this causes the recruitment of an E3 ligase, DCA16, which in itself is undruggable, doesn't have any pockets, it's a quite flat surface. And then these compounds can degrade BRD4 proteins in a very, very, very high potency. So this is a PROTAC, is a glue, you know, it's bifunctional. Like a PROTAC, you can bind two sites, but actually from the same protein. So here to me is one of the examples that is in between. And it's not only DCA16, can be also, the same effect can be achieved with different molecules by recruiting DCA11. So with Amphista Therapeutics, we actually set out to discover novel chemical matter that drives selective interaction between the target and the E3 ligases. And we do this by designing chemical libraries that allow the identification of potent degraders, but because we want to make drugs, we keep an eye from the design already to the properties, molecular properties of the compound, so that we can achieve degraders with oral bioavailability and also CNS penetration. So this approach, because it's not directed to specific E3 ligases, has allowed us to target multiple E3s that can be really recruited to the target depending on the nature of the target and the cellular localization of the target. There's this fly, very interested in TPD, as I see. Yeah, so this is a bit the discovery process that we use. We start from a ligand, from the protein of interest. So we are interested in proteins that are ligandable at the moment, and they are validated in the clinic or with high confidence that they can be very good drug targets. And then we prepare libraries of potential degraders by conjugating the ligand to a series of what we call degradation warheads, although it's a word that we try to remove, yes. So by making these derivatives of the protein of interest ligand, then we assess for degradation in high throughput screening, where what we look at is the disappearance of the protein. We have to be very rigorous in confirming the degradation is specific, is not just due to cytotoxicity, for example. And once we have identified ligands that really warrant multiple studies, then we characterize their mode of actions genetically or with a range of biochemical and genetic technologies. So here I have just a very quick example, and this is a story we've been starting to share quite recently as on the degradation of BRD9. And BRD9 is a very interesting protein, it's a bromodomain protein, which inhibition of BRD9 doesn't basically cause any effect. There is no phenotype by simply inhibiting the BRD9, but by degrading we have quite a lot that can still be discovered, so we're very interested in this. So the ligands have a very nice binding affinity and also a nice solvent-exposed moiety, which we can then derivatize. And by making a lot of compounds, we then identified this very potent degrader for BRD9. You see DC50 subnanomolar and high Dmax. And a matched pair that instead do not cause any degradation. Because, of course, the topic here is quite a lot of covalency. It happens that these compounds are, this compound is reversibly covalent. So you see the cyanacrylamide warhead, while the bulky acrylamide here does not induce any degradation. So I will not go through all the slides, but if anyone is really interested in this story, there is a free online webinar on the 10th of July organized by Tokris Biotechny. So you can just log in and my colleague Scott will take you through all the story. But what we have done, we have studied these molecules really a lot. We identified the mode of actions, and we came out with this cartoon that explains how these compounds work. So yeah, if you're interested in how we arrived here, I recommend to watch that presentation. But basically, we found that our compounds bind with high binding affinity BRD9, and then BRD9 and DCA16 interacts at this. They form a complex, so there must be a protein-protein interaction, probably a natural interaction between these proteins that is not functional, but is probably there. So once the compound is bound to BRD9, and only then, a covalent bond is formed. And the compound glues BRD9 to DCA16. So when the two proteins come together, there is this formation of this new pocket. The compound sitting there, the electrophile is positioned rightly to react with the cysteine, and the compounds glue the two proteins together. So then ubiquitination happens, and then when the target is ubiquitinated, the target dissociates from the compound, but the compound, interestingly, is still covalently bound to DCA16, then allowing a catalytic cycle. So this is another example of how degraders can be really in between protein and glues. So this, to me, is a bit more like a glue, because there is not much interaction with DCA16 alone, but when the complex is formed, there is then the formation of the covalent bond. So these compounds are actually, there are cyanacrylamide, as seen in the presentation before from Matthias. Cyanacrylamide have been in the clinic, and we have demonstrated that these compounds are actually orally bioavailable. They have a good PK, and we could demonstrate that there is an active enantiomer, a less active enantiomer, and the active enantiomer can cause sustained protein degradation in a mouse xenograft. So actually, these compounds are not just tools, and we are doing a lot of work to develop the medicinal chemistry of these degraders. Finally, what is the future? So we've seen induced pharmacology is really promising. It's delivering into the clinic for protein degradation, but also there is so much that we can do by bringing proteins together. So protein degradation via the proteasome and the ubiquitination, degradation to the lysosome and autophagy routes. The ubiquitination has been also demonstrated where you bring together a target protein and a DAB enzyme. Transcription activation, also very interesting if you take a transcription factor close to a promoter, and then steric blocking of protein function. So this is also something that will, here compounds, actually there is already KRAS compounds that are in clinical trials that work by gluing KRAS to cyclophilin. So that really basically causes complete inhibition of KRAS, which is much more potent than what you can achieve with just a small molecule inhibitor. It's been also demonstrated that you can relocate proteins to different cellular compartments by forming these ternary complexes, and also, as mentioned before, target phosphorylation and dephosphorylation. So I would really encourage anyone that is at the start of their career to start thinking about induced proximity pharmacology. Yes, that's me, and I just want to thank all the people that have worked for that project I've shown before from the company, and thank you for the attention.
Great, thank you very much. Thank you, Andrea, for bringing us into the magic of degraders. And now we will finish with the last presentation, if you want to upload your presentation in the meanwhile. So we know that there is a kind of fight. It's like the fight between titans in the drug discovery arena. So there are almost two groups, those who are pushing the small molecules. This would be ourselves, no, the medicinal chemists, those in this room. And also there are people working with the biologics, which are also an important part of the drugs already on the market. So that's why the title of the presentation of Sebastian, I'm not sure if I'm pronouncing well your name, Euler, is it Sebastian? Euler, okay, Sebastian Euler. So he's also coming from a company, Philochem, and I did his PhD with Dario Neri at ETH in Zurich, and now he's working in Philochem, but maybe you will have the slide with everything. Thank you for coming.
Thank you very much. Thank you also for the chance to be here, for the invitation. I'm very excited to talk to you all, to be part of this discussion and roundtable with the title David versus Goliath, Small Molecule Take on Antibodies. I'm talking about the background of targeting, and a bit abstract title because I want to touch a bit on the why and how we could use small molecules for the purposes of targeting. But first, very shortly about me. So as we already said, I'm working now at Philochem. I come from Germany, then I'm a biochemist by training actually. I went from Constance to London and then to Zurich for my PhD under the supervision of Dario Neri, where I worked mainly on DNA-encoded chemical libraries. I will come back to what exactly this is because it's a very interesting tool, also for medicinal chemists. And now transitioned as a scientist and group leader. Group is relative, very small, like we are 3 people working together in Philochem, which is a peer group under the, as group head, we have Samuele Casamali, who actually also thought to come, but he's in holidays right now, so which is my luck, then I can be here to present. And so who are we? Philochem, we are the discovery unit, the discovery center of Philogen. Philogen has worked many years on the use of immunocytokines, mainly for tumor treatments. We have many other fields, and this is based in Siena. And we, the Discovery Center, are based in Zurich, where I'm also coming from. We have both. We have the biology side and we have the chemistry side, and of course the intersection, as we all work at the same side. And so first I want to talk to you about the importance of targeting. So why is it important? And here you can see one slide that illustrates this very well. For example, we have, in this case, a tumor patient that gets chemotherapy, which is typically, for example, in this case, docetaxel, which is intravenously injected, and it pretty much goes everywhere. And after a short time, you see not only in the blood system, but it accumulates in the liver. But, and I can actually tell you, at no time point you really see tumor. It's actually a patient with a lung tumor. And you can imagine this being something that is cytotoxic is bad for 2 reasons. Bad for systemic toxicity, so for side effects, and for the efficacy in therapeutic treatments. And that's why it has been not only in the tumor treatment, tumor diagnostic region, but also many others, but I will concentrate having these 50 minutes on this now, why it has become important to think about targeting. And antibodies actually have been used by themselves, but also because they have, this is a bit more complex with interactions of the immune system and so on, but they have also been used for the delivery of small molecules, for proteins, but I will talk about small molecules as we are here more in the medchem region, and these are the ADCs, the antibody drug conjugates, and by now it's actually an emergent field that now has become very strong. And I just gave you one example. So you have an antibody which binds to, for example, a tumor-associated antigen that then selectively brings payloads that can be cytotoxics or can be also radionuclides for imaging and many more to the tumor site. And for example, here you can see an image of trastuzumab labeled with indium-111, which is a HER2 targeting antibody. And you already see that after several days, you can, already after 1 day, but there you have high background after several days. You can really see some uptake in the tumor lesions, in this case already metastatic. And here you can see a list. I will not go too deep, but a list of some approved ADCs with the trastuzumab deruxtecan. This is so-called Enhertu for breast cancer, which there are also very interesting and promising studies. Interestingly, not only for HER2 positive tumors, we can go into discussion another time, but this is just as an example. Now, why should we move towards small molecules, or what is the interest of small molecules? And I think this slide summarizes it very well. Let's first look at it macroscopically. If you have here a patient that has a tumor just here below the liver and a metastatic patient, and left you have an antibody, FAP targeting, okay, fibroblast activation protein, and you have a small molecule. And you really see the difference. Of course, one also say this is SPECT versus PET, so there's also a technological difference, but antibodies really need a long time to accumulate at the site of a disease, for example, at the tumor site, while small molecules are very rapid, which is good for many reasons and also good for resolution. And you really see this picture here, how in this patient, unfortunate patient, I have to say, you really see all these metastatic sites very defined. And also microscopically, we have actually difference between antibodies and small molecules, so small organic ligands. Again, in this case, FAP, but labeled with a fluorescent molecule. And you can see how upon injection, the antibody penetrates the tumor, not so much because of its size. It makes sense if you think about the size, and you see it mainly around the blood vessels, while the small molecule really very fast goes pretty much everywhere. And that's pretty much why we are interested in small molecules. But as you already know, it's quite difficult to find small molecules that are, for example, ligands against protein of interest. And we have already had an interesting topic, for example, natural products have been also a big source for this. But there has also been the so-called encoded library technologies that have emerged actually originally for antibodies, so antibody fragments by phage display. Smith and Winter that got actually the Nobel Prize in 2018 for this concept, where you couple the genotype with the phenotype, so a phage that carries the information on what it presents, and that allows you the selection. And this then essentially has been more or less copied for small molecules by so-called DNA-encoded chemical libraries, where you have a molecule phenotype that is coupled to a DNA tag genotype. And just very shortly, the history of DELs. They actually are originally from Brenner and Lerner hallmark paper in 1992, so more than 30 years ago now, that just conceptualized that a bit like looking at nature. You have a bead with amino acid sequence, and this sequence you could encode with DNA tags. And this has been taken forward by Dario Neri, the CEO and CSO of Philochem. Also by back in the time, this was at ETH, then led to Philochem and Philogen, and by David Liu at Harvard University to go in solution. And this really established the so-called DNA-encoded chemical libraries that can work in solutions with based on the DNA barcoding, which you can see here. So that you have the DNA that can really encode the molecular assembly that is presented. And this allows the storage and also the screening in an Eppendorf tube, which makes it like, it has a power of like a high throughput screening facility, which is still in use, but also companies that have high throughput screening facilities more and more implement DNA encoded chemical libraries for cost efficiency. And during my PhD, and actually later also as a scientist, I continued this work. I constructed a library based on a proline scaffold, and I don't want to go also for time reasons into too much detail here. But the beauty of using DNA is that you can, the first step, you have to do single couplings, but after the encoding with DNA, you can work and pool, and you do the split and pool procedure. And by this, you can imagine if you have 1,000 and 1,000, you already reach with 2,000 reactions that you can reach 1 million, and this goes on if you even use, for example, 3 building blocks. Or as we did in this case, we used, for example, stereoisomers, which happen to be actually quite important for the productivity of the library. Now, how do you screen such libraries? DEL screening can be done in many ways. One of the most, let's say, established or most commonly applied version is the procedure, the affinity capture procedure in solid support, where you have the library, you have a target of interest that you capture, for example, on magnetic beads, and then by washing, you wash away non-bound library members to have mostly bound library members that can be eluted, PCR amplified, very important benefit also that you have a signal amplification after the selection. And then by high throughput DNA sequencing, you can really start to read and understand. And here you can see fingerprints, very roughly the X and Y axis, the coordinates, they would give you the code identities, so the building block structures in this case, while the color and the Z axis, so in the room, is the enrichment. How often do you see this code combination? So this particular molecule, each point in this case is a library member. And what you aim is to have at the beginning homogeneous count distribution to then end up with enrichments after selections against the target of interest. And this we have done, for example, I give you only one example, which is carbonic anhydrase 9. It's a tumor-associated antigen, which actually we wanted to have isozyme-specific compounds. And we screened CA9 with CA2. There are 14 of them. CA2 was cheap, so that's why we had it. And they're all similar in their active site, or where typically molecules bind. That's why we chose it. And you could really see on the fingerprints you have some similarities, which are sulfonamides. That's common, something expected because they tend to bind carbonic anhydrase. But you also have compounds that are selective for CA9. And these compounds we then took forward. We had a look at them. We did many characterizations. For example, here you can see the SPR profile with a biocore instrument, where you can really see that the compound that we identified, called OncoCA9 now, is specific for CA9, not or very low binding to CA2, and really translated to an exceptional targeting performance, as you can see here with this IVIS image in CA9 positive tumor-bearing mice. It's SKRC52, so human kidney cancer in this case. And now the beauty is, and this is also where we come back to the MedChem, and I think we've already touched on this topic, and I just want to highlight that once you have a ligand available, you can play Lego. You can do many, many things. You can do PROTAC, as we've already heard. You can also combine allosteric with active site inhibitors or binders. You can also couple, in this case, as we have done for this ligand, CA9 ligand, fluorophore for IVIS imaging, which actually can be used. There's Professor Phil Lau at the Purdue University does the so-called fluorescence-guided surgery. Very interesting. There's some nice YouTube videos that if you have a strong stomach, I would recommend you to look at them. They're very interesting how you see that, you really make the tumor tissue glow, which helps to excise it. But you can also couple it, for example, to cages that allow you the caging of radionuclides, as seen here with gallium in a PET image. And this really beautifully shows how this compound in particular went all of it, and this it's a three-dimensional, so you cannot really go around, you cannot lie, you see everything, and you see that most of the uptake is really in the tumor, and that's what you want to have. And we also do the translational work, so actually the compound that I showed you, OncoCA9, we are applying currently for the phase 1 trial in Switzerland, Italy, and in general, so just the translational research, I want to highlight that once you, with the DEL, it's a very efficient, cost-efficient ligand discovery tool. You can find ligands. Then, also using MedChem, you can enhance them. You can play Lego to get different kinds of therapeutic or diagnostic agents that then eventually, hopefully, translate to something that for diagnosis or therapy of diseases like cancer, for example, breast cancer. I mean, of course, there are many types. And yeah, with this I come to the end. Thank you very much for the attention. In case you have questions also for industrial physicians, I would be happy to be here, and I look really forward also to the roundtable discussion later.
Thank you very much. And now that we have so many inspirations, so many hints for our discussion, before starting the roundtable discussion, I want to call Beatriz. Beatriz is an excellent scientist from Spain, but today she's here representing the European Federation of Medicinal Chemistry because she is the treasurer of EFMC. And I would like to, my idea to invite her was not just because we are very good friends, no, not because of that. This helps always in science to have positive relationships, but because the European Federation of Medicinal Chemistry and Chemical Biology, EFMC is the acronym, wants to support all the activities in medicinal chemistry and chemical biology. So that's why I want to hear their point of view about the future. And so thank you, Beatrice, for joining our roundtable.
Don't worry. I had some slides ready, but it was a little bit tight, and so they are not in here. But it's not a problem because there's not much that I'm going to say, just for 5 minutes, as Maria Laura said, and then all I need are some numbers that I have here on my slides, but I can tell them to you, and there's no need to see them. So I'm representing the European Federation of Medicinal Chemistry and Chemical Biology. So you have the logo somewhere here in the so I know Gianluca was on Monday presenting the European Federation, but I know he didn't mention a few figures that I'm going to tell you now, which is that the EFMC represents 31 societies from 26 European countries and close to 10,000 medicinal chemists and chemical biologists around Europe. Of course, its mission is to support the medicinal chemistry and the chemical biology community in Europe. What I wanted to highlight from European Federation is that it also has a global reach worldwide with relationships with Canada, United States, Brazil, of course all over Europe, and China, India, Australia, Japan, Korea, and we maintain these relationships constantly with different events and different scientific exchange. But there are two relevant things I wanted to highlight today in relation with the future of medicinal chemistry. One of them is that in 2019, the European Federation for Medicinal Chemistry founded the Young Scientist Network. And this Young Scientist Network, I won't say anything about it because Michele, who is somewhere in there, I know he will be talking tomorrow. So, but I think this is a very important moment in the history of the European Federation for Medicinal Chemistry, the foundation of the Young Scientist Network, and this is obviously looking to the future. And the other important change that has occurred in the EFMC is that, and this is probably the only slide that I would have liked to project, but it's not so relevant, is that originally it was EFMC, European Federation for Medicinal Chemistry. And in 2020, there was a change, slight change in the logo, not very relevant, but a very significant change in the name, which is European Federation for Medicinal Chemistry and Chemical Biology. So I think that these two facts, the incorporation of young scientists that has been followed also by other countries, like in Spain, the young scientists had also created a group. I think in Italy, you were here meeting the other day, and I had to escape because it was not my meeting. And so I think it is clear from the EFMC what is the point of view, where the future is, which is in you. It's also in the international relationships, also in the relationship with pharmaceutical industries. This is another slide that I cannot project, but all the, not only the sponsors, but also the participating in all the activities. And here today we also have a very important representation of 2 companies. So I think the message is clear from EFMC. So thank you very much.
Thank you, Beatrice, for these nice words. Yes, and now we have time until 5 p.m. for our discussion. So I would like to truly thank you for sending your questions. So I will start, and the questions are from people from the audience. But then, as I said, I would like maybe that the answers from you will be very short, so maybe we have time also for other questions from the audience here. So I will start by talking with the question about covalent inhibitors, because yes, it has been the topic of the first day. We have reasoned a lot on that, but still some of you had already something to clarify. And the specific question was about covalent inhibitors, and if covalent inhibitors could provoke mutation onset. And then the question continued, could we overcome these by reversible inhibitors? So I don't know who wants to start, maybe Matthias, Chiara, the 2 of you, to give some comments on this. Thank you. Maybe you, Matthias?
Yeah, I mean, or if Chiara wants, as you prefer.
No, but please go ahead, because
Okay,
you are
then
from.
I go for it.
You are
So,
from the outside. You know that in Italy we care a lot about hospitality. So you're coming from abroad. So that's why I always start from those abroad.
Okay, great. So yeah, I mean, that depends a lot on what you're trying to treat with a covalent modality. If you go for a cancer indication, you put selection pressure on your cancer cells, and this provokes resistance upon treatment. But it's the same for reversible and irreversible inhibitors. What makes the difference is for irreversible or a covalent inhibitor, what often happens is that the target amino acid mutates. And this is something that you can overcome either by selecting a target where the target amino acid has a function for the protein, which can be bad because then you may lose selectivity because it's often conserved. Or alternatively, you can try to make a ligand which has already a very high non-covalent binding affinity and has the covalent mechanism on top, which means even if it loses the covalent mechanism, that it still has a high affinity, and the covalent mechanism is then there for enhancing PK/PD properties, target residence time, etc. If you have a non-cancer setting, that's a different story. There you don't have the selection pressure, and there usually you also don't see this problem of resistance formation towards covalent inhibitors.
Thank you very much. Chiara, I want to hear from you as well.
Yeah, sure. So I think Matthias really covered
Yes.
almost everything. So maybe what I can add that if we have resistance to covalent inhibitors, we could also try to explore a broader surface on our protein, so we can try to go on different sides. So for example, make this bivalent orthosteric allosteric compound or go only for allosteric compound. And sometimes, if we have mutation, we could also try to exploit different amino acids. And now we have all chemistry in hand, because they have been shown, for example, that we could covalently target also aspartic acid or arginine, that since a few years ago, it was impossible to have warheads targeting this kind of amino acid. So we have really a lot of opportunity to really overcome the resistance.
Okay, thank you very much. Once again, I want to stress a concept which is my favorite one. Nowadays, I want to invite all the community to not use anymore the word warhead, especially in these days. So if you want to replace this term, I would like to invite all of you to do that. So because you are those, it's very common, we know, it's something, but worried has also another meaning, so that's why maybe we could change our language, and Andrea already agreed with that. So thank you, Chiara. And so then there was a question about multi-target compounds, about the pro and cons of multi-target compounds and how to rationally design them. We didn't talk specifically about polypharmacology and the multi-target approach, but maybe again, Chiara and Matthias can say something because there are multi-target kinase inhibitors, so maybe you can. Okay, I want to hear from you. Maybe I can add.
Yeah, I can be quick, then I leave the word to Matthias. So I think it's a bit related to what we discussed the first day, that the fact is what you want to do with your molecule. So if you want to make a drug, there are actually many drugs that they work, and they are multi-kinase inhibitor, and they do work because they are multi-kinase inhibitor. So if you want, for example, to treat a disease and the drug works, this is not a problem. But on the other side, if you want, we discussed about chemical genetics, so if you want, for example, to use your compound as chemical probe, then you need to be very selective, so then you can't use a multi-kinase inhibitor. So I think it really depends on what you want to do, and also when you are designing the compound, you really need to know what you are looking for.
Thank you. Very nice clarification, Chiara. Very nice point you made. Matthias?
Yeah, I mean, I think, yeah, I fully agree with Chiara. You need to distinguish between chemical probes, which you use for mechanistic studies, and drugs. If we now look more at a drug setting, I mean, there are a variety of reasons to go for multi-target compounds. For example, many diseases are not driven by one exclusive target, and if you can target several proteins involved, then you may have a better effect. And also, along the same lines, you may have a chance to prevent resistance if you attack at several sites at the same time. The problem is rather how to make these compounds. And if you have targets with a very similar pharmacophoric pattern, you may be able to really design a classical small molecule which fits into both and is still relatively selective, or even into 3 or 4 proteins, but is selective against many others. If you can't do that, then you can do bigger molecules which have one side fitting the one protein and the other side fitting the other protein, but then you may also end up with having something like a molecular glue, of course. And if you make it even bigger, you can make hybrid compounds that are a bit similar to PROTACs, where you have one side targeting the one protein linker, other side targeting the other protein. But of course, that kind of compounds are much, on the one hand, easier to design, but on the other hand, also more difficult to develop due to their size and their likely limitations in terms of bioavailability, etc.
Yeah, very nice comment. I agree 100%, Matthias. So to me, the lesson is this one. So it's multi-target compounds can have advantages, but they are very difficult to make and even to rationally design them. So if you want to go for a multi-target drug discovery project, you should have very clear since the beginning about the true advantage of doing that. And to demonstrate, you can do, for example, you should understand whether the 2 targets that you want to hit, they are interconnected. Why I'm saying that? Because if they are interconnected, then you can have synergy. Otherwise, please, don't go for multi-target drug discovery approach because they are so complicated to make. I can say this because I was working in the field for more than 20 years. So very nice, there is strong rationale for multifactorial disease, but please start from the right targets. So don't embark in a very difficult project without having a strong rationale. So I don't know. What do you think? I say people here saying yes, yes. So everybody from here agrees.
Can I make a comment?
Yes, please.
You just explain the golden rule of drug discovery, probably. That anything you do, there has to be a very strong rationale for doing that.
Yes, this is true. But especially in this
yeah,
case.
and degraders as well. It's so hard to do it, that, you know.
Very true, yes, Andrea. You are perfectly right. Okay, so now let's go to the other question regarding, let's switch to the natural products, so similar. So there was a question related to expanding the sp3 chemical space. So is this a promising direction? What will be the role of a CSP2, CSP3 cross-coupling? Maybe Luca is the right guy to start the discussion on that, and then we can jump in.
Yeah, I'm happy to jump in there. Yeah, definitely. So, I mean, yes, it's been shown in several studies over the years that sort of FSP3 tends to increase, you know, throughout the various phases of sort of preclinical and clinical development. So there is a kind of a push to build this in from the start. So people like myself and others who are interested in designing compound libraries for screening and sort of hit identification do try and factor that in. I would say though that it's three-dimensionality. It can come obviously from an increase in FSP3, but some let's say some molecules that look quite flat on paper can actually still have quite a significant element of three-dimensionality. And also it very much depends on what kind of target you're after. So there will be binding sites that are, you know, flatter by nature and would thus be more suited for those kinds of compounds. I would have thought that sterile binding proteins would be one such site, but it turns out that they are also a little bit more three-dimensional than we anticipated. So I think overall I would say it's something that we certainly factor in our design of libraries and explicitly factor that in, and particularly in the inclusion of things like spirocyclic compounds that give this sort of inherent three-dimensionality to the kind of compounds that we make.
Thank you. Yes, I believe that we have learned that we should escape from the flat land, you know. So, okay.
Indeed, yeah, I didn't mention that in the talk, but I think that's certainly something that the sort of the proponents of this approach are very keen to highlight.
Thank you. So now let's go into the products and degraders. And so there were several questions. One was related, these are for you, Andrea, of course. But there was one was related to the difference between PROTAC and molecular glues. But I believe that we could skip this one because Andrea already explained very well the difference and even all the nuances in between. So it's much more complicated than just this division between PROTACs and glues. But then there is another question: how challenging is applying targeted protein degradation to infectious diseases, Andrea?
Okay, yeah, good question. I've never tried, so but I can share perhaps some of the difficulties that one can encounter even just working on new E3 ligases. So I guess if you want to target E3 ligases in parasites or mostly parasites, I guess. You would probably have to use different chemistry because you may not want to target necessarily the human E3 ligase ortholog. There might be a difference in the chemistry that you have to do. You have to discover new ligands, and discovering new ligands for E3 ligase can be very difficult. They can be difficult to drug, but also when you have a ligand for an E3 ligase, it doesn't mean that will work in inducing protein degradation. That's because E3 ligases are highly regulated. So sometimes you have the E3 ligase is there, you have your ligand that binds to the E3s, but nothing really happens. And that's because really there is a lot that we don't know. And then I think there is a huge challenge on the practical point of view. For human proteins, if you want to degrade human proteins, there is quite a lot of antibodies that you can find. You can do Western blot, right? If you degrade proteins In a parasite, I don't know, many have antibodies that are ready to use for Western blot, so you probably have to invest a lot in the detection methods and assay development. So yeah, overall, I think is a challenge, definitely.
Yeah, maybe, I don't know if it's also your point, it's better to go for antiviral products because in this case we are exploiting the human machinery. So what do you think?
Yeah, definitely. So I think there is some work ongoing and probably something published already on this. So yeah, if you can exploit the human machinery to target a viral protein, that's probably a better way, definitely. So yeah, also in bacteria work is quite interesting. So you know, there is no ubiquity in bacteria, but there are proteases that have been targeted. There is a work of a dear friend of mine.
Yes.
Italian, yes. Francesca Esther Monreale, she's at the Crick Institute, and she really pioneered the ligands for CLIP-C as a bacterial proteases. So, but again, I know by talking to her also in bacteria, you know, to look at protein degradation, now they have to do mass spec because there are no antibodies to follow protein, or you need to tag proteins, and then you really devise some very engineered systems. Yeah.
Luca, do you want to say something on that?
No. Actually, I was just gonna comment on the same thing that you just mentioned, that the really nice work on the back PROTACs.
Yes, very nice. I agree.
That's very cool work. But I agree with everything that's been said. Definitely looks like a challenging avenue.
Thank you. And so another question on related to degradation. So you have to start. Maybe we can even ask Sebastian, maybe, because this is something more peculiar. We didn't touch RNA so far, which is another hot topic. But then the list of the participants to the round table, we would have need all the day for the round table. We didn't touch RNA. But we know that RNA is a hot topic, and even ribotac are hot topics. So these are specific degraders. So I don't know if you want to make a comment on that.
Yeah, again, not direct experience for me on that.
Okay.
But I know people that work on that, and the work of Matt Disney probably opened the way there on targeting RNAs. I think, again, the difficulties, from what I understand, is really to find good binders for both RNAs and really RNA, which probably people expect it to be a bit easier than it actually is. I like to see what companies do, a bit because of my job also. I find it quite interesting to try to gather as many information from websites and so on. And there is a company called Arrakis that really pioneered the targeting of RNA, and they were founded just a couple of years before us, and from my understanding, they're still really working hard on their own targets. So you know, you may find a specific RNA that you think is a very good target, but then again you develop all the chemistry and then nothing happens. So is that because the chemistry is not good enough then in vivo, or is it because there is again something that we miss? So what I find really interesting, perhaps, is although again it's early days, is the use of nucleotides in general to recruit transcription factors to E3 ligases, for example. You know, we could think about these chimeric molecules that have nucleotide that recruits the transcription factors to an E3 ligase, and then we can degrade those.
Sebastian?
Maybe a small comment add-on from my side is that, like, targeting RNAs, I think there's the difficulty of that you have, also if you think about silencing and so on, so you have to have a certain length that you have to target, otherwise there are too many off-targets because it would repeat in other sequences, and I think this makes it challenging. And what I found also interesting, of course we have seen now with the vaccines on RNAs become important in many ways. In the silencing field is actually that they try to couple it to either by formulation, that they try to target delivery, which is also still a bit of a difficulty. And there have been some small molecules that have been used for DNA and RNA delivery, which is still really early, and difficult by transport proteins, or with like kind of nanolipids or structures that include some targeting elements. But this I see also as a big challenge.
Yeah, yes, selectivity, it's truly a big issue, yes. And now maybe we can come to the $1 million question. So there was, this was the question. The head of a big pharma told me, I skipped the name of the big pharma, okay, but the head of a big pharma told me that all biotechnologies available today will make medicinal chemistry extinct. What is your opinion? So you have to start. This is, as I said, this is the question, the question. You, Sebastian. No, because you talk a little bit on antibody drug conjugate, which are something in between, and also.
Well, I think, of course, if you look at biologics, there have been big progress, and I think there are 2 points of view there. One is the research part, and one is actually the legislative part, because if we start from the legislative, I see the problem if you look at the USA, for example, that they have enforced the law of price control on small molecules, not on biologics, so that companies start to move away from medchem, from small molecules, also because there's such a price pressure, while with producing biologics, you can charge up to 1 million and nobody says anything. And I think this is already a wrong direction, and I don't really see, they always argue for the purpose of production costs, which if you look at the research costs are not always the high factor in that case of drug development. So this is, I think, one important point to consider, and at least in the US, I see it going into the wrong direction. While then, for the pricing part, I also see that medchem and small molecules have a big benefit, that we will be able, not only pricing, so it could be much cheaper to produce it. If you think about drugs that are biologics or that are small molecules, we do both, so I really see the cost comparison, and it's quite significant. Yeah, I think that's pretty much, now I lost my last point, so I hand over and maybe I come back to that one.
Okay, maybe I can add that, yes, price is a big difference, but we don't want to, we should not just cure the developed society, we should think to cure all the world. And so I believe that we cannot do this with biologics, but we cannot neglect a big part of the world where it would be impossible to cure diseases with biologicals. And so this is something to keep in mind, especially for me, people like me that work in academia. So I should consider that even the ethical consideration when we approach drug discovery. So I believe it is so important. If you remember the case, as Sebastian mentioned, of the vaccine and its availability in Africa during the COVID, we discussed a lot about that, so we should have this in mind, okay, regarding even the access to medicine, that we should ensure, for sure. I would say that small molecules in this sense are more popular, in the sense that they are more approachable to everyone. Do you like my definition of small molecules, like popular? It was just created now. Okay, so is anyone else that wants to add something on this topic? Otherwise, I will move on.
Maybe just very shortly.
Yes, Matthias, please.
So, so 2 things. So, I mean, I have some colleagues that keep telling me since probably 20 years that medchem has no perspective and it's not going to continue. And at the moment, I rather see medchem again on the rise than on going down. That's the one thing. Second thing, if you talk with people from industry, they often look at things more from economic perspective. And if you look from an economic perspective, it's clear that biologics make a very big part of the cake. But if you just look at, for example, approval numbers, the situation is already different. And third thing, I think that the borders are getting less and less clear. I mean, what does a medicinal chemist do? A medicinal chemist is also necessary for making things, for example, like antibody drug conjugates or things like peptides, modified peptides, also modified proteins. And these are all things that also belong somehow in the domain of medicinal chemistry. I think the scope is just getting broader and more diverse, but I don't see that medicinal chemistry is that, no.
Thank you.
Yeah. Can I just say, I fully agree, and I would also like to say to the other panelists that I think both Andrea and Sebastian, I actually use kind of your two fields as examples when people ask me, you know, is medicinal chemistry dead? Well, I say, look, you've got Protax, that's a hot modality, that's not going away anytime soon, it's only growing. And same with these sort of small molecule conjugates that the Sebastian that you presented, and I've seen Dario present, several times. This is a beautiful example that I can sell to people and say, look, medicinal chemistry is even more important these days to make these very complex small molecules. And so being a synthetic or medicinal chemist is, it's not going away anytime soon. I fully agree with all the points that Matthias made.
Yeah, absolutely, Luca. And so a topic, we received a couple of questions on these topics, so I collected all of them and make a general question that we can share among us. So the importance of green chemistry and sustainability in the medicinal chemistry of the future. And first, the importance, and second, how could we integrate it into our daily activities in the lab? Who wants to start? If you want, I can start, because I'm very sensitive to these topics, and especially I liked how this question was formulated, because there was green chemistry, and this is a problem, of course, but green chemistry is around since many years. No, it's not something new. There are the 12 principles of green chemistry and so on, but then the question was referring to a broader concept, which is the one on sustainability. And I want to draw your attention that sustainability, we use the word sustainability for many things, okay? And even we know very well the Sustainable Development Goals by the United Nations, but let's think about sustainability from a medicinal chemistry point of view, and especially talking about drugs. And again, sustainable means not only that the compounds that we are producing should not affect the environment, should not affect the ecosystems, and should not be toxic, but sustainable for us means, again, what I was saying before. Sustainable means even considering the problem of access to medicine, which is very important, very pressing nowadays. So when you talk about sustainability, yes, it's green chemistry, but there is another side of sustainability in drug discovery, which is related to the access of medicine. So I'm very glad that this was considered by you, by our PhD student, and also how we can integrate these in our daily activities. We are very lucky because we have, from the strict point of view of chemistry, we have the 12 chemistry principles, no? And we should stick to them. We should consider them, no, when we are in the lab. So use less toxic solvents, reduce the number of steps. They are very, very nice. Some of them maybe do not fit very well to drugs, but I want even to draw you to consider, for example, one of the principles related to biodegradability. No, there is one, because these 12 principles are for all the chemicals. And then there is one related to the fact, I'm looking to Antimo because he is very much involved in this. And so when you think about a drug, should the drug be biodegradable? Yes, it should be biodegradable, because we know that when the drugs are then in the waste stream, then they are toxic to the environment. This is not easy, but it's something that we should start to consider. But we should not be too shocked, because I remember very well when I was a student, Jurassic Park, so no. Yes, but when my PhD was devoted to find the most active and the most selective molecules at our times. Then we understood that the PK was so important. So we shouldn't be scared. Maybe we should even reconsider this property for a drug, which is biodegradability. I'm not saying that it's easy, but I'm saying it was not easy at all, even considering including PK into the drug design and the drug development process. But now we are quite good in that. Or even before the problem of toxicity, no one was considering before ourselves toxicity. But our generation, we were always testing toxicity. And so, yeah, something's changing. I don't know if you want to say something, otherwise we can ask even the audience here. But to talk, you have to come here because this is the only microphone working. Yes.
Maybe I can just add one point. So yes, I completely agree. I think the problem is also sometimes we don't know yet which are the groups that can't be introduced on the molecule in order to make them not to involve the degradation in the environment. So for example, we have seen some papers recently where they mention about the CF3 and CHF2. I have to say that I put during my postdoc on all over the molecules because of course these have excellent properties to enhance the blood-brain permeability. So sometimes I think it's something we really have to balance, considering the toxicity for the environment and the properties that these moieties gives to the molecule.
So, do you want to make a comment? Do you want to come here to say something? No? So maybe just to talk about practical action, everyone can contribute to a green chemistry approach. For example, why don't you get rid of notebook and you just use an electronic notebook? This is something very easy to do. We can start even from small things every day. So don't use notebook and use electronic notebook. Then maybe we can even discuss about the use of more computational studies before making the right compounds. I'm not saying that I'm always for the experimental proof, but of course, if you can reduce the number of molecules to make with a proper computational study, this is also very good for the environment. Then even to reduce everything, all the micro approach, where we reduce the solvents and everything. There are several ways, but I believe that we are in the right track, no? And so, Antimo, do you want to come here, say something, maybe, to add to the discussion? Oh, all of you, I don't know. Because I was looking to Antimo, that was...
Yeah, maybe I can add
yes,
a bit
yes,
of...
Matthias, please. Antimo.
If it's okay? Or
yes.
I can also wait.
No, it's your turn.
Yeah, I mean, I want to add a little bit of controversy to that because on the one hand, of course, it's a very honorable goal to make drugs that are biodegradable. But on the other hand, we always should consider that this adds another layer of complexity on top. I was discussing about that a lot. I didn't mention that I also made a 4-month internship at BASF after my chemistry diploma, and there I was in crop science, and there biodegradability in soil was always a problem, and it's not so easy to optimize. And with respect to what we are doing in the lab, so in my perception, I mean, of course, whenever we can reduce the amount of consumables, things that go out in the environment, it's a good thing. But if we do medchem, we also have to keep an eye on efficiency of our processes. In our early research, we need to be quick. And I think then it can be counterproductive to put too much effort in keeping every synthesis route that you develop green, because, I mean, if you can directly start from scratch with a green method, that's fine. But putting additional effort in a research route that is green is probably not so useful, whereas it becomes very, very relevant once you go for a process route where you need to scale the whole thing up. So I think these are two different sides of the coin. Yeah.
Yeah, that's why I was making a comparison with the PK parameters, because at the beginning, we, and Beatrice is agreeing with me, when we were listening, oh, PK, we should consider all these optimization to have a molecule, but now it's something which is routinely done. So that's why I'm not saying that tomorrow we will be able to make biodegradable compounds, but if this is the direction, maybe we can work on that, not now. Of course, efficiency is always, as you were saying, so important, because the pharma companies, they are companies, so they cannot waste money, of course. But in any case, especially maybe for us at the academia, we can start to think how to approach the problem. I'm not saying that should be into the pipeline very soon, but to me, this reminded the same concept of PK that was not too much into our drug discovery when we started. So that's why I was making this kind of comparison. But I agree, it's a further layer of complexity that we should deal on. But in any case, I want to stress that the only call by the European Union dedicated to drug discovery was devoted to greener pharmaceuticals that are biodegradable. This was the only call dedicated to drug discovery a couple of years ago. Do you remember, yeah. Antimo? So this is something that we should consider at least.
Yeah. So I would like to shift a little bit the attention on another point of sustainability that in many cases is not considered in our lab, which is the sustainability of our work, of what we are doing on our lab, and the sustainability of the project in terms of efficiency in the way we conduct the project. Because as scientists, many times we just think to do things and not to think before doing things. And this is especially important for the people working in the lab that often suffer the pressure of doing things because the idea is always to have you know, the good results. So sustainability is not only something that means how we perform the chemistry, but also how we treat the people in our lab. So I think this is something that in Italy always is not considered. So I think this is something that we need to think about. And also the way and the place where we work is important. So I think that we have to offer to our students the best place to work, because working in a nice place is a plus. And I think that this is an attention that we have to care with, because finally we are here as senior because of the people working on the bench. And I think that the first sustainability that we have to think about is this, in my opinion.
Please stay here. Everyone is clapping. Yes, thank you for highlighting this. Very nice. Thank you for sharing this with us. Yes, we should never forget that, and we should try always the best for the next generation. And so basically this was also the the reason behind this roundtable, which is going to the end on time. So if there are no further questions, do you have the very last moment question? No, you are a bit tired. So yes, with following your words, I believe that we wanted to demonstrate here with this roundtable that we, as the ESMEC School, care a lot about you. We are convinced that you are the future. We want that you stay in the field, that you take care of our discipline, as I said at the beginning, so I want to close as I started, so that you share your best ideas and you try to do this, I hope, in your country, because this is also another phase of sustainability, so of the how you can do your work in a sustainable way. If you can do in your country, will be even better. If you want to go to another countries, even better. I don't know, as you prefer. But yes, I hope that with this roundtable, we have provided you good examples with these very brilliant young investigators that with so much energy have inspired us and give us some hints on what to do and how to do it. And please always remember that there are also the scientific societies. And so now I'm talking as a president of the Division of Medicinal Chemistry. So we are here to support you. So consider that. And even EFMC, as I said before, EFMC is there to support everyone. Here you are coming from all over Europe. And so all of you belong to the EFMC. And also maybe what we should also highlight is that we are here all together, and even joint efforts is so important, and networking is also so important, especially for a multidisciplinary science like it is ours. So that's why I'm so grateful to the people who have participated here to this, to me, fantastic roundtable, and coming from different countries, from different topics, but very willing to share their ideas and their suggestions with all of us. So thank you very much, and I close the roundtable. Thank you. So maybe we make 5 minutes of break. Where is it? We can go. Okay, so let's resist a little bit. Let's move to the last part of there is the last part of the program. And this is dedicated to something that you should be interested in. I don't know if you didn't look at the program. We are now at its 4th edition. We have received several applications, but I'm very happy to share with you the recipient of the 2024 edition, who is Professor Adriano Mollica. Please join me in welcoming Adriano Mollica. So, look, how do you like this? Very nice award. So, next one could be yours,
Only
okay?
For Alumni Award.
So, this is for you, Adriano, with our best congratulations. Picture.
Evviva. Moment. Also this for you.
We have everything.
Many things to
Many things,
take,
yes,
you
many
can put,
gadgets, so very nice award.
Okay. Grazie.
So, I'm not going to say too much about your career, because maybe you are talking a little bit, but Professor Adriano Mollica is professor at the University of Chieti, and is very well known because of his study, medicinal chemistry, on opioid ligands. His science is mostly devoted to peptides, but also to small molecules. He was trained also in the lab of Victor Ruby in the US, who was one of the father of opioid peptides. And during his career, Adriano has continued to be involved in the area of natural products, because he's also working about the development of nutraceuticals. And so I'm very eager to listen to your talk. And again, congratulations, Adriano. Presentation, please.
Okay, thank you very much, first of all, to the board of ESMEC to give me this important award. It's really an honor for me to be here. So I want just to... Yes. Yes. So I found this picture. It is me in the school in 2005. I was at the third year of PhD, and I was just came back from the United States. So I presented this poster in black and white, you can see, about my early work on opioid peptides. So today I decided to start from that point and show and share with you some more recent work about opioid peptides. So, as we know, there are three main opioid receptors: mu, delta, and kappa. And these receptors are involved in the antinociception. The mu opioid receptor is the most studied, and morphine and its derivatives bind to this receptor. This receptor is also responsible for the side effects. So side effects like the development of resistance, tolerance, the respiratory depression, and so on. So we are trying to develop novel agonists. Also, the other opioid receptor involved in antinociception, but with less side effects. So we are trying to develop mixed agonist. Let's start to the natural endogenous opioid peptide, such as enkephalin. So enkephalin is a pentapeptide, and it has this peculiar structure. The first residue is a tyrosine, then there are 2 glycine, and the 4th residue is phenylalanine. Then there is a 5th residue that can be methionine or leucine. Later have been discovered other opioid peptides, more complicated, like dynorphin, beta-endorphin, and these peptides are longer, but they share the same initial sequence. So the first amino acid is tyrosine, the second is a glycine, the third another glycine, and the 4th is phenylalanine. Then lately have been discovered also endomorphin 1 and 2 that are mu-selective ligands. And also these peptides have in common the first amino acid, tyrosine, the 4th amino acid, phenylalanine, and then they have 2 other different residues in position 2 and 3. So we can understand that the first and the 4th position are the opioid, represent the common part, so the opioid motif. Okay. So, since we have already many opioid agonists, why we are still researching for novel agonists? Recently have been found two new features about opioid receptor. The first is called the biased signaling. So the G-protein coupled receptor, once activated, can activate a second messenger response, mediated by G-protein or mediated, for example, by beta-arrestin. Ok. Credo sia finita Zoom. Vado avanti, ok. So, if we can modulate this response, we can avoid the side effect. We know that the internalization of the opioid receptor is due to the recruiting of beta-arrestin. So if we can divide these 2 activation of the receptor, we can have only the antinociception without the side effects. Another observation, very recent, is the properties of the opioid receptor to form dimer, homodimers, or even heterodimers. So there is the new possibility to design molecules that bind to the orthosteric binding sites or to the allosteric binding sites formed by the heterodimers or the homodimers. So, starting with the natural ligand, why we can't use in therapy this natural ligand? Of course, these ligands are not selective and are very flexible, and they have a low bioavailability, they don't cross very well blood-brain barrier, they have a low metabolic stability, and so on. So we need to improve the structure, to improve the stability at least, and design the peptidomimetics. During the years, been developed a hierarchical approach to design peptidomimetics. So, as a first step, we should identify the biological active peptides. Then we have to study the structure-activity relationships to understand which residue are the pharmacophore. And then we start to consider also the three-dimensional structure of the peptide and modified this three-dimensional structure, applying some constriction, so such as cyclization, or even local constriction on the single amino acid. So, following this hierarchical approach, I based my studies on opioid peptides. So, some of these modifications have been done several years ago, starting from the leoenkephalin. One of the first modifications was to change the second residue with a D amino acid in order to gain some metabolic stability. Later it was discovered that the fifth residues can be eliminated, and after this the Polish scientist Lipkowski used enkephalin and make bifalin, which is a double enkephalin. It has a symmetric structure, and the two branches of leoenkephalin are joined by an hydrazine bridge. So I was intrigued by this structure, and I started to modify this lead compound. So one of the first modifications I did was to substitute the hydrazine linker with the other bisamine. And I found this compound that I named AM94 with piperazine, and also I used a fluorinated phenylalanine. So this compound was much better than bifalin. The binding is sub-nanomolar, and it binds to delta and mu receptor. Also is very efficacious to activate the receptors, the G protein, and you can see that the antinociceptive activity measured after intravenous administration is very good and very higher than bifalin. This is because it is more stable and crosses better the blood-brain barrier. So I moved to another modification that was on the backbone by substituting the alpha amino acid with a beta homo 3 amino acids. It's very close, the beta homo 3 amino acid, to the natural amino acid, but it has a methylene bridge, an additional methylene bridge on the backbone. So usually this modification is always detrimental for the activity, because it changes a lot the freedom of the conformation of the peptide. So in fact, I tried to substitute each residues, and only one substitution was tolerated. There was the one with the beta homo 3 phenylalanine. So the peptide containing this residue was able to bind to delta and mu receptor in nanomolar range, and also we measured the degradation in human plasma, and we can see that the product containing the beta amino acid is much more stable than bifalin, and also it is more active in the in vivo studies after intravenous administration. In this graphical, we can see that in red is morphine. So morphine after intravenous injection is always more potent than these peptides. This is because it crosses better the brain barrier, and also it is more stable. Then I decided to understand, to discover if the bifalin can bind to the receptor in a folded conformation or in a linear conformation. So I decided to cyclize this molecule. And to do this, I substituted the residues in position 2 and 2 first with cysteine. the cysteine was protected by a protecting group, the acetamidomethyl protecting group, and after constructing this linear peptide, by oxidation in methanol with iodine, I was able to create this disulfide bond and make the cyclization. So I tested this compound. We used L-cysteine and D-cysteine, and we discovered that only the product with D-cysteine was active and was able to bind and stimulate the mu delta opioid receptor, whereas the product containing L-cysteine was barely active. So this is an important finding, I think. After this, we improved the structure using the penicillamine in place of cysteine. This is because was found before on this other peptide, DPDP, that the use of dipenicillamine instead of cysteine confers a delta selectivity. So we want to try if this is can be applied to bifalin. So we synthesized the model, and once again we discovered that only the product with dipenicillamine was active and able to bind strongly to delta mu receptor. The product containing L-penicillamine was not active, but we didn't have a great success to target only the delta opioid receptor. Indeed, this product is slightly selective toward the opioid receptor, delta opioid receptor, but still very potent because we tried the in vitro test with several in vitro tests, and we discovered that the product is much more active than morphine in intrathecal cerebroventricular, the intracerebroventricular administration, in the hot plate test and the telephilic test, and after intravenous activity, instead, again, we have a good activity, but morphine is much more active. Then we moved to the cyclization by dicarba analogs. The dicarba analogs, the double bond. And this can be a more stable product. Also, there is the possibility to modulate the conformation by having a cis trans product. So to obtain these two analogs, we used the Grubbs catalyst, a second generation Grubbs catalyst. This was invented by Robert Grubbs, that is a Nobel Prize received a Nobel Prize for this invention. So this catalyst. So we used this, and we were able to obtain the cis and trans isomer. In this case, we had a surprise because there is a little selectivity for DOR, but these two analog are not able to stimulate the G protein related to DOR receptor, the delta opioid receptor, but only to the MOR. So indeed, they are agonists to the MOR receptor and neutral antagonists to the DOR receptor. Still, the antinociception is very good, better than bifalin. One of the last modifications was using the CLIPS technology. So CLIPS is the acronym of chemical linkage of peptides onto scaffolds. It uses the reactivity of the two cysteine groups, two thiol groups of the cysteine, by reacting with dibromoxylin, and is able to make this kind of cyclization. It is very versatile because it can be used to do bicyclic, tricyclic analogs, and also to attach a fluorescent probe. So we used, I skipped the chemistry because it's very easy, but we used all the isomers of dibromoxylin So we were able to create 3 analogs with different sides of the cycle, the 26, 27, and 28 members. Then we tested again for binding. We found that the best binding was about the product 8 with the smallest cycle. We tested also for anti-nociception. We found that was a great improvement of anti-nociception after intrathecal administration. So we decided to join this latest modification to another modification that is called the modification in the key space. So the key space is the space determined by the free rotation of a side chain of amino acid. So it must be considered because there is not only the overall conformation of the peptide, but also the positioning of each side chain. So it has been found that using, for example, a dimethyl tyrosine, it can be useful for modulate the activity of opioid peptide. So we generated several mixed analogs with different linkers, with different type of cyclization, and with the dimethyl tyrosine in place of tyrosine. And actually, we find that the most active compounds are the linear one, so MJ1, MJ2, and also are interesting MJ4 and MJ5. But the most active are these 2 linear peptides. And if we can have a look to the anti-nociceptive activity, we can see that these peptides are very, very stable and very potent over 4 hours. So this is a very, very good data, I think. So I conclude. So far we have designed several bivalent analogs with non-hydrazine linker, with beta-amino acid, cyclic analog by several type of cyclization. So at this moment we are able to have a better blood-brain barrier penetration, stability for over 4 hours, activity by intravenous administration. But of course, the challenge is to create compounds that don't recruit the beta-arrestin, so we don't have the side effects. And actually, we are working on this. We are measuring all these compounds that we made for this. So, thank you very much to your kind attention. This is my group at the moment, and all the other collaborators.
Thank you, Adriano, for this overview on your work, and especially it was nice because we didn't touch too much peptides, no, and now again peptides are rising at the moment, and so Adriano introduced some interesting concepts like the clip technology and macrocyclization, which are also new direction for medicinal chemistry. So this was good. To finish in this way, so I hope that you will have a lot of food for thought. And thank you for staying here so long. And thank you to all the participants, to Adriano, again to the organizer, to Beatrice, to the organizer here, and the scientific committee, Marco Macchia, the director, and see you tonight. Thank you very much. Bye.
Sorry. Tomorrow morning. Sorry. Tomorrow morning there will be Dr. Tiziana Bandiera for the Market Watch. So we will meet tomorrow just at 9 o'clock. Okay, please try to be here. Okay, thank you very much. Punctual at 9 o'clock because it is a very interesting lecture. Thank you.